Hello dear students, welcome back to my channel. Today we are going to learn about facilitated diffusion using interactive fat simulation. So let's start. In the previous video we have learned about simple diffusion. Here in facilitated diffusion, unlike simple diffusion where tiny molecules like where oxygen or carbon dioxide slip right through the bilipid layer, some molecules like sodium ion, potassium ion, which are charged, they need some help, they need little help, they need a doorway. That's where this membrane proteins come in. Let's start. First of all, the membrane. In the center we have our cell membrane, the phosphobilipid layer. Above it is the outside of the cell, below is the inside. If we see the membrane potential, notice the plus sign and the minus sign. At this point, outside is positively charged and inside is negatively charged. We can actually see the exact membrane potential on the right side, that is minus 70 millivolt. On the left we have our solutes. Here on the left we have these solutes. We can add gases like oxygen, carbon dioxide, sodium ion, potassium ion, and larger molecules like glucose. The channels on the right is the star of today's show. The protein channel, here you can see, these acts as bridges across the membrane. These, I'm talking about these on the right side. We have three types: leakage channel, here sodium ion and for potassium ion. We have voltage gated channel for sodium ion and potassium ion again. We have ligand gated channel, sodium ion for potassium ion. Right. Now let's start the demonstration. We will start with the leakage channel. So these are like doors that are always open. Here, that are always open. The yellow channel only lets the sodium through and the blue only lets the potassium through it. Watch what happens when I increase the concentration of sodium and potassium ion on the on outside of the cell. So here I am going with the sodium ion first. I am increasing it, and let's play, and let's see what is happening. Yes, you can see here they are going with the going through the sodium ion leakage channel, and at the same time when I increase the concentration of potassium ion on the outside of the cell, the blue one, let's see whether they are able to go through the yellow one. No, they will only go through the potassium ion channel, the blue one, right? Because this is a passive transport, the molecules want to move from higher concentration to the area of lower concentration. Here higher concentration is outside the cell and lower is inside the cell. Watch them carefully down their concentration gradient through the leakage channel into the cell. No ATP is required here, so it's a passive movement. Here you can see the solute concentration of sodium ion and potassium ion. Now let's look at the voltage gated. We'll send it back and we'll move to the next channel, that is voltage gated channel. These doors aren't always open, they have a lock, and the key is electricity, specifically a change in membrane's electrical charge. Right now At minus 70 millivolts, the orange sodium channel is closed. You can see here, right? And let's move the sodium ion. The sodium ions are bouncing off it. Yes, here you can observe. But if the signal causes the inside of the cell to become more positive, a process called depolarization, let's slide it to this minus 50. Let's see. Boom, the channel changes shape, it opens up, and sodium rushes into the cell. This is exactly how nerve impulses travel through your brain and body. Now finally we have the last one, ligand gated channel. So we'll send it back and we'll see the ligand gated channel here. Ligand is just a fancy biology word for the chemical messenger like neurotransmitter or a hormone. Notice this channel is closed, right? This is closed. Even if there is huge concentration gradient, the ions can't get through it. Let's increase the concentration gradient of sodium ion outside the cell. Watch what happened. The sodium ions are not able to go through it as the ligand gated channel are closed. Now when I click to add ligand here, these little molecules are binding to the receptor site on the protein. This binding causes conformational changes here. This binding causes conformational changes here in the protein. If I show you again like this binding causes a conformational changes. It changes the protein shape, forcing the gate open so our ions can flow through it. Now to summarize, facilitated diffusion is passive transport. It moves molecule from higher concentration to lower concentration without using cellular energy, but it requires a membrane protein to act as pathway. Whether it is always open or whether it is always open leakage channel here like this, or electric sensitive voltage gated channel, or a chemical sensitive ligand gated channel, these proteins are vital for keeping our cells functioning. So here you can see that's it. Here on the screen you can see I have put all the 3 types of channel on the membrane, and yeah, that's it. Through leakage channel, which is always open, sodium ions are passing, but not through a voltage gated channel and a ligand gated channel. So that's it. We have learned about facilitated diffusion, how these channels facilitate the diffusion of ions through the membrane. This is what we have learned. I hope this video helps you to understand this concept of facilitated diffusion. Thank you very much.